some advisor about this one, morphologic and digestive tract changes of hatchery rare tomato clownfish and pepper apprentice. I'm so sorry because actually this is not related to agriculture and some of you not so understand about it, but I think this is because our cluster is agriculture and agribusiness. So I think we can take from agribusiness area. So maybe some of you Taiwanese or Indonesian like to be businessmen, especially in ornamental fish. So I think this paper can be very useful for you to information or conduct some, some experience. So today, this overview. So I just investigated on morphological development. So if some of you like to watch a movie, My Nemo, My Finding Nemo. So very nice movie. We can know from the movie how they have their habitat, how they socialize with some others in the video, and how they can breeding and something like that. So maybe this paper can explain how the baby development. So up from the egg to the pre-larvae. So we can explain here morphological development, growth, and digestive tract of this amphibian apprentice larvae. As your information, in the Finding Nemo movie, it is not this species, it's another species. We call it amphibian ocellaris. So this is another species because we have around 28 or 29 species about this one. As you know that if the result can be very useful for businessmen, especially for ornamental favorites. They can manage the larvae rearing, and also for scientists to ichthyophysiology studies. So this is the objective, concern on morphology and growth, and also developing the digestive system. So this is why it's very useful because we can know the development of this system. We can know when we can feed some kind of feeding or some kind of diet. As you know, ornamental fish, especially from salt waters, cannot be artificially propagated. It just be naturally propagated. So natural breeding. Even we cannot do any artificial breeding for this species. So this species is very famous now. As your information, our country Indonesia has many islands and I think many coral reef because this is the species associated with coral reef, and also this one have very nice color. It's called colorful, and also well adapted in aquarium condition. So as we know that this is the gray, this is the yellow sign. This is like distribution of this species. As we know, Indonesia has include. But if you read the information, which country that have a high production of ornamental fish? Do you know? Could you guess the country?
Singapore.
Singapore. How come? They are a very small country, but we are a very big country. They high production of ornamental fish. How come? Even they don't have any coral reef. You see? So this is very question tag for us. So this is have 29 clownfish species, and then this is just justification to enrich information about my research. And I already told you, and this is some literature review. This is species, Yeah, this is species. In Finding Nemo, we can find other species have two white stripe here, stripe here. But this species has only one, and also famous in Malaysia when I pursue my master degree. This is some what we call classification, and this is methodology from general rearing conditions. So the problem of this breeding is because this natural propagation is a little bit difficult to stimulate their propagation. So how I, with my professors, some have secret recipe to stimulate their breeding condition because we cannot inject them. We just stimulate by feeding. So we already take some oyster spirulina, something that we combine. So we got secret recipe. Can we stimulate them even every 2 weeks? They can have egg. So this is some secret recipe. So and then after we success to breed the fish, and then we have the what we call the larvae. So we take sampling for 15 larvae per every day, until 20 days. And then I draw. It's very difficult, 1 millimeter small larvae we draw. Very detailed about morphology, but I did it. And histology procedure and sampling for histology, as we know before about embryology, but Miss Justina, she also applied some histology process and some data analysis. So this is larva rearing. So we can see here, maybe some of you not so familiar with this, I just skip and just take a picture. This is recirculation system. This is the tank. We have 24 tanks here, and every day we monitor the water quality, and also we feeding here, artificial feed, artemia, siphoning, and etc. And then histology procedure. Maybe some of you very familiar with this. Very boring working, right? And then data analysis, just regression, because it's development. Yeah. So for development, we just use regression. And result discussion. This is some kind of picture, right? But this is drawing. We can see here some of morphology here. So we use a Lucida microscope drawer to draw this larvae. And then this is 1 day after hatch. As your information that they have incubation egg for 8 days for this species. So first, the what we call, after we stimulate the maturity, so the female will put the egg in some place, like some substrate, like in the bottom or in the pipe, and then the male will follow the female, and then will put their sperm. And for 8 days, the incubation, the egg will be released from the substrate, especially the bottom or what we call pipe. So this is 1 day after hatching, 2 day after hatching, 2 day after hatching. Maybe you're not so familiar with this, some kind of term, primordial fin fold, and then this is PF. But I just wanna put you some information. This is like the process, how the Notter chart occur, how the fin occur, and how the stomach occur. And this is until 6 days after hatching. And you see here some what we call this is melanopore. Melanopore means black spot. So when the fish still baby, they don't have what we call the stripe yet. They just have like this one, melanopores. And then by 7 days after hatching, the chromatopores will be appear. That means like oranges or reddish color in the fish. Okay, this is until 10 days after hatching. You see here, melanopores were replaced by chromatopores fully at 8 days after hatching. And then white band, you see white stripe is already appeared in 10 days after hatching. It's very interesting to see, wow, finally I wait for 10 days to appear, because how come from the baby they can appear the white band like that? This is already some better combination from drawing. And then this one until 20. And it's quite interesting, for 20 days after hatching, it's appeared another white band here. Actually this species just has 1 white band, but this white band will be disappear in the next step. Okay, so this is the growth feeding, the growth pattern until 20 days after hatching. And then just feed some rotifer and green water, Artemia nopli, and some secret recipe here. And this is the real what we call larvae. Yeah, from this one, very small, 1 millimeter at least, more or less, and until this is 1 day, until this G, this is like 17 days. You see, yeah, with double white band. This relative growth from compared to total length and other parameter, and it has very significant relationship. And this one, some histology section, even in the 1 day after hatching, it appears some digestive organ already developed. YS here, mid-gut here, gut and liver is already, even it's just 1 day after hatching, amazing. Because another fish, they just 1 day, maybe they just have still yolk sac or something like that, right? Yeah, thank you. What's going to happen? Okay, this is the 2 days. It's already some organ already developed. Hindgut, midgut, stomach, esophagus, and this one. And this is 14 days. Is some already developed, fully developed. And this is 17 day after hatching. It's already good, have a good organ and can digest the diet very perfect. And the conclusion, we can see it here, that this amphiprentice hatch at the post-larval stage with fully developed and functional eye, mouth and fins. The commencement of pigmentation, like melanopore, started from pre-hatching and then replaced by chromatophore, and then the appearance of white band along the body and head. The total length has significant relationship with standard health and height, body depth, and eye diameter. And this histological of digestive tract of this species show the larva has straight tube closed at the mouth and histological differentiated along its length as newly hatching. And also found some several functional organs such liver, midgut, stomach at 1 day after hatching. It's still baby but already found. Therefore, live foods such as rotiver are suitable to be fed at early larvae after hatching. Thank you for your attention.